Okay, the next thing we'll talk about is proteins and absorption of proteins within the intestinal lumen. First, the proteins are broken down by pepsin inside the stomach. The hydrochloric acid starts denaturing the proteins, and the pepsin starts breaking the bonds of the polypeptide bonds and breaking them into smaller polypeptide bonds. Once they are smaller, they go into the lumen of the intestines, and they are broken down into smaller and smaller peptide bonds. They're broken down further smaller and either into very small peptide bonds or amino acids. The enzymes that break down these polypeptides within the intestinal lumen are called brush border enzymes, and they continue to break down amino acids into smaller components. Once they are broken down into amino acids, dipeptides and tripeptides, they go into the enterocyte. Once again, they cannot enter the enterocyte as polypeptides. They can only enter it as amino acids, dipeptides or tripeptides. Once they're in this formation, they're able to enter the enterocyte via the help of active transport secondary and facilitated diffusion. The amino acids and the dipeptides enter the enterocyte via active transport, secondary active transport. Remember that active transport requires ATP, active. If it's active, it uses energy. Remember that for the MCAT, active transport requires energy. However, facilitated diffusion never requires any energy. It's just from a high concentration to a low concentration gradient. It's going downhill, down current. Remember, diffusion, don't let it trick you just because it's facilitated. Anything that's diffusion does not require any energy. Therefore, it enters into the enterocyte. Once it is in the enterocyte, it leaves. It's converted further by enzymes, intracellular enzymes in the enterocyte, and it leaves in the basolateral side of the enterocyte into the capillaries and to the hepatic portal vein to the liver.